The title of my project is The Expression and Localization of 3Beta-Hydroxysteroid Dehydrogenase type 1 protein in human breast cancer cells. Breast cancer is correlated to a variety of factors, including steroids and breast composition. The focus of my project was on steroids. In human breast cancer cells, there has been seen to be much higher levels of the steroid progesterone as compared to regular breast cells. This steroid is synthesized by 3-beta-hydroxysteroid dehydrogenase protein in steroidogenesis from pregnenolone. Of this protein, there are two types in the human body, type 1 and type 2. Type 1 occurs in the ovary, which regulates breast growth, which is why I studied type 1. In this project, I tried to compare the expression of this protein in human breast cancer cells transfected with the DNA for this protein as opposed to non-transfected breast cancer cells and monkey kidney cells. I also localized the protein and checked the complexes with which it's associated. I predicted that there would be much higher expression in the transfected breast cancer cells than the non-transfected, and that there would be very little to no expression in the monkey kidney cells, as per my research. To conduct this experiment, I subcloned the DNA in a vector which I used to transform bacterial cells, from which I isolated plasmid DNA and used these positive clones to transfect my cells, which I grew, then lysed and isolated the protein, and used Western blotting to measure expression. Later, I translated this protein and incubated with mitochondria and ER to see which one it was imported into. I found that there was much higher expression of this protein in transfected versus non-transfected cells, and that there was little to no expression in the monkey kidney cells, as expected, and that it was imported into both mitochondria and ER, but associated only with the mitochondria, but only associated with the mitochondria, not integrated into the membrane, and associated with the ER, but not integrated into the membrane. I then attempted to determine the complexes. It is currently ready for mass spec. From the results of this project, we can conclude that 3-beta-hydroxysteroid dehydrogenase type 1 protein is much more highly expressed in breast cancer cells, transfected and non-transfected, than in monkey kidney cells, in which there is very little expression. We can also see that 3-beta-HSD1 is imported into mitochondria and endoplasmic reticulum and associated with both, but not integrated into either. This suggests that there is a protein group with which 3-beta-HSD1 binds on the mitochondrial and ER membranes. This may be because there is partial modification of the targeting group, and thus it is targeting to two different organelles showing high expression. In future, I would like to identify the additional factors and determine the degree of post-translational modification.